I'm going to try. I really want to run this off of my iPad today. So we're going to see how it goes. Get to that. Hopefully. Well. Okay. Oh, and I'm supposed to do. A couple of y'all forgot to click in for attendance on the last time y'all were here. And I put myself a sticky note and I wrote y'all names, but then I can't find my sticky note. So if you were here last time, please just tell me. Yes, send me, yeah. Just say I was on Zoom. Oh, oh. Fine. Okay. So don't forget to do iClicker. Okay, so then I need to join Zoom on this. Let's do that. Okay. Fingers crossed. This works. All right, let's see. It's been like so long since I've seen y'all. Did I tell y'all the farmer joke? No? Okay, how does a farmer play music? He turnips the beat. It's cute, right? All right, let's see. Why was the baby strawberry sad? Because it found out its mom was in a jam. It's a little dark. It's a little bit dark. There were some other ones that were really good. I don't remember. Two's good.
I have one for you, Dr. B.
Wasn't feeling very well. It's very good. It's very good.
Dr. B, I have
Very
one.
good. So don't forget, y'all, check in on iClicker and send me an email. If you didn't check in last time, there were like 3 or 4 people who didn't check in last time. And I wrote it down and then I lost the sticky note. So I don't know. I don't remember. So just send me an email that says, hey, I forgot to check in. Okay. All right, Sam, what's your joke? Yeah, I think everything's up to date. Okay. What's an owl's favorite soda? Hoot beer. Hoot beer. That's good. Hoot beer. We like that one. We like that one. All right. So I'm going to try my iPad because I really love using it. I'm such an iPad user, such a Mac user. All right, let's try this. Yes. Woohoo. All right. So, lipid metabolism, right? We ready for this? All right. So, all of chapter 16 is degradation and synthesis pathways of fatty acids, right? Our lipids classify under fatty acids, triacyl glycerols, and cholesterol, right? So we're going to talk about synthesis and degradation of all three. Okay. Yeah, it's going to be great. So the first section is fatty acid oxidation, and we'll also talk about ketogenesis. Section 2 is synthesis of those fatty acids and the triacylglycerols, and then section 3 is all cholesterol. So it's a nice breakdown. So here's kind of the big picture. You kind of have to have the big picture to start with, right? So here's our metabolic map. So what do you notice about the metabolic map? Where do we have a key substrate? Where are all the arrows pointing? Acetyl-Coa. Acetyl-Coa is where it's at. So acetyl-Coa is the primary link between our fatty acid metabolism and the energy conversion pathways, right? So when we have amino acids coming in, you haven't eaten in more than 3 months, right? What's your body going to start doing? Breaking down proteins, breaking down whatever it can, right? Making acetyl-CoA. What if you just ate lunch and you got high glucose levels? What's going to happen? You're going to metabolize the glucose into pyruvate, which then into acetyl-CoA, right? Okay, so what if you go on a low carb diet, right? What's going to happen? Your body's going to start metabolizing the stored fats, the stored triacylglycerols, and turn them into acetyl-CoA. Exactly right. And what's the other thing that we've already kind of talked about how we make cholesterol? What's one of the precursors for cholesterol? Acetyl-CoA. Oh my gosh, right? Acetyl-CoA is also a precursor for ketone bodies, which we'll talk about in ketogenesis. So acetyl-CoA is really our key player here. So, oh, I'm going the wrong way. All right. So we have to be very careful about regulating. Where's my mouse? We have to be very careful about regulating degradation and synthesis of fats, right? And then once we decide, okay, if we're going to undergo degradation, we're going to go fatty acid oxidation. What happens in fatty acid oxidation? We're going to take those fats and we're going to generate acetyl-CoA, right? So we break down the fats and we're going to generate lots and lots of acetyl-CoA. And then what's it going to enter? Go back one. What's it going to enter? If we take fatty acids, we turn them into acetyl-CoA, then that acetyl-CoA enters what? Citrate cycle. So like we're going to have to recall all that stuff from the citrate cycle, right? Okay. So the opposite is true. If we consume too many precursors of acetyl-CoA, that acetyl-CoA is going to be converted into fatty acids. And then we can export them, we can store them, that kind of stuff, right? So when we talk about responsible fatty acids being responsible for providing long-term energy, how long? What are we talking here? Like if you go a couple hours without eating, is your body going to revert to fatty acid degradation? No, what are you going to revert to then? Stored glycogen. Stored glycogen is going to be short term. So you have to go kind of a while until you start actually breaking down fatty acids. And then how long is it going to last you? About the average person. How long do your fatty acid stores last you? About 3 months. About 3 months. Remember the 3-3-3, right? You can go 3 days without water. You can go 3 months without food before you start breaking down like muscle mass. It's 3,3. I don't know what the thing is, but something like that, right? You can't survive long without water. You can't. Like 3 days. About 3 days.
It might be 3 minutes without oxygen.
That's what it is. 3 minutes without oxygen. That's what it is. Look, Sam's got us. 3 minutes without air, 3 days without shelter and water. Yeah. 3 months without food. 3 months without food. Yeah.
But if you're like working out and burning your glycogen, like if you're doing a fasted workout and you burn your glycogen stores, would that speed up?
Yeah, that would absolutely speed it up. So absolutely. Oh yeah, yeah. Well, and all these numbers are basically on the average person who's doing normal stuff, right? Not on a massive workout, not already on a diet, you know, so all of those things would factor in, and then you would get a different metabolic rate depending on what's going on, right? Okay, so if let's say we eat too much, we're going to store excess acetyl-CoA. Where are we going to store it? Right, we say store there. Where are we storing it? We're going to store it as triacylglycerol, sure. Where are we going to go with that triacylglycerols? Adipose, ADIPO, adipose. And what else? It's going to be, so muscle is going to be glycogen. So you're actually going to store some in the liver, which can be a big problem. We'll see. We shall see in our case study. All right. So when we talk about fatty acid metabolism, there are 4 key enzymes that you have to know and understand how they work. All right. So the first one is fatty acyl-CoA synthetase. So the first thing I like to talk about is what is the difference between a synthase and a synthetase? Anybody know? So originally, all synthetases consumed ATP in their synthesis reaction, but synthases did not. It is not always true, but it is a good rule of thumb that if you have the 2 T's, it's going to use ATP. So that's the difference between synthase and synthetase. So the fatty acyl-CoA synthetase, this is going to basically prepare the fatty acid for metabolism. So what it's going to do is it's going to convert free fatty acids into fatty acyl-CoA molecules. And this conversion happens in the cytosol. Where do you think we need to get our fats in order to metabolize them? Where is the citrate cycle? In the mitochondria. So are we in the mitochondria? We are not in the mitochondria. So you have to kind of think about cellular location wise, where are things happening and what's going on. So after we prime it, then what we have to do is we have to transfer it from the cytosol to the mitochondria, right? So we have carnitine acyl transferase 1. This is one of the enzymes that is required in that transport system to get it from the cytosol to mitochondria. And this catalyzes this enzyme. It's not the only enzyme in the pathway of transferring from cytosol to mitochondria, but it is the most important one because it is rate limiting. What do we know about rate limiting steps? Another word for rate limiting is irreversible commitment steps, right? So we're committed. Commitment step, right? We're not going backwards from here. So we'll be able to transport our fatty acid into the mitochondria so that it can be processed. Now, this relates to fatty acid oxidation, right? So oxidation, you always want to think about as what? Breakdown or build up? Degradation, breakdown. So like that should automatically be one of those things that you automatically look at and say, oh, oxidation, that's breakdown. But just like we talked about gluconeogenesis and glycolysis, right? We have competing pathways. So Acyl-CoA carboxylase, this is the rate limiting step in fatty acid synthesis. So these two are participating in competing pathways, right? So for the carnitine acyl transferase 1, this one, so this one right here is inhibited by a substance called malonyl, L-O-N-Y, malonyl-CoA. Malonyl-CoA is the product of the Acyl-CoA carboxylase reaction. Product of CoA carboxylase. Does that make sense? That the product of the fatty acid synthesis enzyme, rate limiting enzyme, inhibits the breakdown. Yeah, if you're synthesizing it, do you want to break it down? No, not yet, right? And that makes sense. So we have to kind of always be thinking about regulation. So when you look at the acetyl-CoA carboxylase reaction, this one can be regulated both by reversible phosphorylation and allosteric, all those allosteric mechanisms that we always talk about. So phosphorylation, ION, and allosteric, A-L-L-O-S-T-E-R, allosteric mechanisms. So just like most other enzymes that we've talked about so far, right? Okay, then we have fatty acid synthase. So fatty acid synthase is what, once we've primed it, basically in order to create the fatty acid, then we're going to have to keep adding and making these fatty acids longer and longer and longer. So these are going to add carbons in 2 carbon units. So what you'll notice about most fatty acids is that they're even numbered in terms of the number of carbons inside of them. And it's because fatty acid synthase adds them 2 carbons at a time. Okay. Yeah. All right. So we're going to talk first about fatty acid degradation. So when we talk about fatty acid degradation, the first thing that you have to do is you have to prime that fatty acid. So how do we say you prime the fatty acid? You attach it to an acyl-CoA, right? So then just thinking big picture, not on here. So you attach it to the acyl-CoA, and then what do? Right? It's kind of like a 2-step thing. Prime it, but it's primed in the, what part of the cell? Cytosol. So then transport, okay? So priming is what's going to occur first, and that's going to occur in the cytosol. So when we talk about priming it, right, then we're going to add carbon units to it. And you have some enzymes that will add carbon units that are when the molecule is really short. Then you have some that will add carbon units when the molecule's of a medium length. Then you'll have some that will add once it's really long. So you really have a specificity of synthetases depending on how long the fatty acid chain is. And you don't have to know the names of all of them. You just know the general group. We're not getting that detailed in it. So we have a 2-step reaction that's going to happen, and it really helps to look at the mechanism. Okay, so we want to form a fatty acyl-CoA. So we are going to link this to ATP hydrolysis. Okay, so we're going to start out and we have a fatty acid. So how do we know we have a fatty acid? What kind of a functional group do we have on the end of the fatty acid? A carboxylic acid or carboxylate, right? Depending on the state of the cell. In this case, we have a carboxylate because the hydrogen is not there. All right. So the carboxylate ion is what is going to attack our ATP molecule. So when you look at ATP, right, where are the high energy bonds? Between the phosphates. So remember how when we were in Biochem 1, we were like, okay, this is our alpha phosphate, our beta, and our gamma. Remember that? Okay, so our fatty acid is going to attack the alpha phosphate of ATP. So when it does that, what's going to happen is that you are going to add one phosphate group with adenosine attached to our fatty acid. That leaves you with, what did you just release? Remember, we're going to think about energetically, how are we going to drive these reactions to happen? We're going to couple them to ATP hydrolysis, but how does that actually work? You had to put in energy to break a bond, so we didn't actually do anything energetically favorable yet. What's energetically favorable here? What did we produce? Pyrophosphate. What happens to pyrophosphate? Turns into inorganic phosphate with a very high negative delta G value, right? So if you look down, we have a high negative delta G value. So that makes that part of the reaction energetically favorable. Now we have an intermediate, right? So our first reaction here was by the fatty Acyl-CoA synthetase, right? So we've attached this one phosphate group with our adenosine, and now this complex, this intermediate is still bound to the enzyme. The enzyme hasn't let go of it yet. So we're going to have a second reaction where the thiol group, right, there's the thiol group, right, is going to attack our fatty acid. So when that happens, we're going to release the AMP, right? So AMP is lost. This whole thing is going to be lost, turns into just simply AMP. And then our thiol group is going to attach to our fatty acid. So now we have a fatty Acyl-CoA, and this is what gets released. And so that release of the AMP is going to generate a relatively large negative delta G value. And that's what's going to drive our reaction. Okay. So it's a two-step mechanism in that very first fatty Acyl-CoA synthetase. Good. Yes. Very quiet today. It's tough back from a break. A little bit. Yeah, a little bit. Okay. So what did you have to invest to make this happen? You had to invest in ATP. So just kind of keep in mind, like keep running in your brain how much energy you're producing, how much energy you're consuming, right? Because we're going to want to keep track of that at the end. So in order to do this, you had to consume an ATP, and then you have to remake the ATP. So it actually kind of costs you 2 ATP in order to do this, because now we're going to have to go back and regenerate that ATP. All right. Okay. So now, what happens to our fatty Acyl-CoA? Well, it really depends. You have to look at the energy charge of the cell. If the energy charge is low, what does low energy charge tell us? Have we eaten in a long time, or has it been a while? It's kind of been a while, right? Okay, so if it's been a while, we want to move that fatty Acyl-CoA into the mitochondrial matrix. If we do that, we need a transport system, because it's not just going to diffuse across the membrane. So that's going to be called the Carnitine transport cycle, and that's what we're going to talk about next. So once we put that fatty acid into the mitochondria, what we're going to generate are a bunch of Acetyl-CoA molecules, a bunch of FADH2, and a bunch of NADH. What are those substrates for? Acetyl-CoA is a substrate for citrate cycle. FADH2, what does that do? Electron transport chain. NADH, electron transport chain, right? Yes. So if our energy charge is high, we've eaten, we're not really hungry, right? So we're going to favor fatty acid synthesis. But we're only going to favor fatty acid synthesis, right, depending on what's going on in the cell. So we don't want to import fatty Acetyl-CoA into the mitochondria if we're synthesizing fatty acid, right? So we're going to produce, when we undergo fatty acid synthesis, we're going to produce malonyl-CoA. And we haven't gotten to that yet, so we'll get there, right? But that malonyl-CoA is going to inhibit degradation, okay? All right, so let's talk about, we've made our fatty Acyl-CoA. Let's assume that our energy charge is low. So now what's the next step? What are we gonna do? Bring it in, bring it in. So we're gonna do the carnitine transport cycle. So this is where we're regulating whether we're going to go through degradation or synthesis, right? So one thing that's really cool about this is that we're going to decide, do we want to degrade the fats into real energy, right? Our energy charge is really low, or do we want to make them and we want to use them for the membrane, right? So if we decide we want to degrade them, that's great. But acetyl-CoA, our coenzyme A, is super, super important in both the cytosol and the mitochondria. So if you bring all of that coenzyme A into the mitochondria, you're not going to have a cytosolic pool and you're going to be in big trouble. So basically what happens is that when we get to transporting this acyl-CoA molecule, we're going to release the acyl portion of it, the coenzyme A, let it go, transport it in, and use a mitochondrial pool. So it's going to keep a separate pool in terms of cytoplasm versus mitochondria, right? And so this is also controlled by malonyl-CoA. If we have lots of malonyl-CoA, we're synthesizing all of these fatty acids, and so we're going to inhibit transport into the mitochondria, right? And that's what we want. So let's look at this mechanism. So here's our transport cycle. We're going to take it one step at a time. We got 3 different steps. So we're going to take just a general fatty acid, palmitoyl-CoA. So this is going to be the fat that we're just going to kind of talk about as we go through this reaction. So we need 3 proteins and we need a molecular tag, which is called carnitine. So this is our fatty acid. You can see the long fatty acid tail. You can see that you have your carboxylate, and then your carboxylate is hooked on to carnitine. Okay, so this is our tag that's going to allow us to transport the fatty acid in and out. So when we come, what are we bound to? We're bound to the thiol group on CoA, right? So what do we have? We have an acyl-CoA. So what we're going to do is we're going to go through the very first step as our carnitine acetyltransferase 1. Our very first step is catalyzed by this enzyme, and it's this big thing right here. And what are we going to, we want to keep a separate pool of CoA for the cytosol as we do for the mitochondria. So we have to let go of the CoA, but we need something else bound to it. So can we have free fatty acids? No, because they'll disrupt what? Membranes. So we can't have that. So we're going to put this carnitine tag on it. And once we put this carnitine tag on it, we're permeable through the outer mitochondrial membrane, but then when we get to the inner mitochondrial space, we can't get through. So you have to have a transport system. So you have the carnitine acyl carnitine translocase, and it is a transport protein that will pull our palmitoyl carnitine into the inner mitochondrial matrix. But at the same time, we're going to ship a free carnitine out. So what kind of a transport protein? Antiporter, right? So we're going to bound one bound to the fatty acid is going to come in, and the free one is going to come out. So the third step, right, is we can't metabolize our fat in this state. It's not going to work. So we need to do the reverse of what we did in the cytosol and just swap that tag out and put a CoA back on. So now this is mitochondrial CoA that we're going to swap on and tag that onto our fatty acid. So we basically have the same thing in the cytosol that we now have in the mitochondria.
Really could go through the inner mitochondrial membrane, but you need that translocase so that the carnitine can come back out.
No, you're not going to with a carnitine tag on it, you're not gonna go across the membrane. You're not going to. Mm-mm.
So the carnitine helps it go across just the inner membrane, right?
Correct.
So it can go through the outer membrane without the carnitine.
Correct.
So what is the difference between the two membranes that allows it to be?
It's a different phospholipid makeup.
They're both like hydrophobic. So like
They are both hydrophobic. They are both hydrophobic, but just because it's some small differences, but it's going to allow it to go across that outer membrane, but not the inner membrane. But you don't have to know the membrane differences. Like don't get bogged down. Okay, so we have our fatty acid, right? We have it linked to CoA and we're in the inner mitochondrial space, right? So now what do we have to do? What's our next step? We have a really long fatty acid. We're gonna have to chop it up, right? What do we get when we start out with a long fatty acid? What are our products? We talked about this. We're gonna make Acetyl-Coa. We're gonna make FADH2 and NADH. Okay, so turning a really long fatty acid would be helpful if it would be on the screen, right? Into these little pieces, right? How many carbons are in an acetyl-CoA? Yeah, right? It's 2 carbons. So what are we going to do? We can't have a really long fatty acid. We're going to have to chop it up. So when we chop it up, this is what we call our beta oxidation reactions. Now, I'm going to show you a little animation just because I want you to remember how we talk about carbons in the chain. So when we number a fatty acid, where do we start the numbering system? At the carbonyl carbon, right? So this is basically carbon number 1. I don't know if it's going to let me write on it. Carbon number 1, good. Then the next one is carbon number 2, then carbon number 3, carbon number 4. But there's an old system that if you took carbon 1, this is the carbonyl carbon, right? So it already kind of has a name. So then they said, okay, well, the following carbon, which we now call carbon 2, is the alpha carbon. Okay, now the next carbon, right, go down the alphabet, alpha, beta. So we are actually going to end up cutting these 2 carbons off. And that's why we call it beta oxidation, because we cleave it between the beta and the alpha carbon. Okay, so you just kind of have to know there's eventually what will be cut. But we're going to go through all the steps, but I want you to kind of see the animation first. But I want the sound to work, so I'm going to go through and I'm going to take over on here. And I have it pulled up. Aha. Okay. Thought I could make it big, yes. All right. Well, it would help to have the volume ready.
Level of a cell is low.
Sorry. I'm gonna, hold on. Okay. Let me just back it up. All right, so this is going to give you an overview of all the different reactions that will occur in order to break down the fat. Okay, so it gives you a nice overall picture before we start talking about all the details.
Degradation, beta oxidation reactions. If the energy level of a cell is low, fatty acids like palmitate are oxidized to yield a substantial amount of energy. Fatty acid oxidation occurs inside mitochondria. To enter the mitochondrial matrix, fatty acids are first acylated by coenzyme A. The coenzyme A group is then temporarily exchanged with a molecular tag, carnitine, that marks the fatty acid for transport across the mitochondrial double membrane. Once inside the mitochondrial matrix, carnitine is replaced by coenzyme A, and the fatty acyl coenzyme A conjugate molecule enters the beta oxidation pathway. The beta oxidation pathway has 4 steps. In step 1, the beta carbon single bond is oxidized to a double bond. The electron pair extracted from the single bond is transferred to an FAD-bound enzyme, which reduces FAD to FADH2. Eventually, the electron pair is transferred to coenzyme Q, which contributes to the production of ATP in the electron transport chain.
So do y'all remember, I'm gonna back it up just a little bit, when we were talking about, oh, you gotta get it just right.
Contributes to the
Okay, so do you remember when we were talking electron transport chain? And one of the things we listed was ETFQ oxyreductase. Do you remember that? And I was like, yeah, that's fatty acids and degradation, right? And y'all like, okay, sure. It was just one of those things you listed. Well, that's where it's going to come in. Okay, so that should be kind of be a nice flashback.
the production of ATP in the electron transport chain. Step 2 of the beta oxidation pathway is a hydration reaction, with a water molecule being added across the newly formed double bond. The alpha and beta carbons of the fatty acyl coenzyme A molecule are shown here. In step 3, the resulting hydroxyl is oxidized to a carbon-oxygen double bond, and its electrons transferred to NAD+ to produce NADH. Lastly, in step 4, acetyl-coenzyme A is cleaved from the end of the fatty acid. The fatty acid, now shortened by 2 carbons, is ready for re-entry to the beta oxidation pathway. Each subsequent cycle of the beta oxidation pathway releases a 2-carbon unit and 1 molecule each of FADH2 and NADH. So how many rounds of the beta oxidation pathway are needed to degrade a 16-carbon fatty acid like palmitoyl coenzyme A?
How many? 7.
The answer is 7 rounds, producing a total of 8 molecules of
Total of 8
acetyl-coenzyme A
molecules.
and 7 molecules each of FADH2 and NADH. The energy conversion reactions of oxidative phosphorylation inside the mitochondrial matrix provide a total of 108 moles of ATP for every mole of palmitoyl coenzyme A that is completely oxidized by the beta oxidation reactions.
So the animations are really nice. I don't know if y'all have been going back to watch the animations, but a lot of times, a lot of those mechanisms where you're showing a stepwise, where they show a hydration, they show whatever it is, the animations are really nice on it. So I thought I'd remind y'all that way that they're really awesome. Okay, so now I'm gonna switch back. I think I'm gonna switch back. Yes. Yay, we're back. Okay, cool. All right, so where are we doing beta oxidation? And why do we call it beta oxidation, right? Now you know why we call it beta oxidation, right? Because that's the carbon where you're cleaving between the alpha and the beta carbon. Okay, so where are we? In the matrix of the mitochondria, So you can add in matrix, right? Okay, so we're going to degrade our fatty acid 2 carbon units at a time. And the overall process, we call this thiolysis, right? So what are we generating? Y'all told me FADH2, NADH, Acetyl-Coa. And so every time we go through this process, it's 4 repeatable steps, right? 4 steps in order to do this. So what are our 4 steps? The first step is a dehydrogenase or an oxidation reaction where we're going to lose our hydrogens and we're going to form a double bond. Now, it's actually really important that it forms a trans double bond. If it were a cis double bond, we're going to have problems, and I'll show you why later. But just note that it is making trans bonds, and you'll find out why that's really important later. And this is also isoform dependent upon the number of carbons in the chain. So you have multiple Acyl-CoA dehydrogenase enzymes. Some enzymes work if the molecule is really long, some work if it's medium length, and some work if it's short. Okay, so it's isoform dependent, but you don't have to know extra names and all that kind of crap. Okay, it's enough. You got to draw the line somewhere, right? Okay, so then we're going to do the hydration. So if it's a hydration reaction, right, we have water. Water is going to break apart. The hydroxyl is going to go on one of the carbons, and then the hydrogen goes on the other carbon. Anybody remember from the animation which one's which? As it across the new double bond, what was the new double bond? Watch, let's do this real quick. In that first reaction, where are we creating a new carbon-carbon double bond? Between the alpha and beta. So this was our original double bond. It's not a carbon-carbon double bond, it's a carbon-oxygen double bond, right? So the new one is between alpha, and beta. Okay, so if you want to go in and you want to add that to your slide up here, right? Between alpha and beta carbon, right? So then we're going to add the water across that new carbon-carbon double bond between the alpha and the beta. So what gets what? It's stereospecific. It's really stereospecific. So here's our second one, right? We're going to hydrate. So what gets what? Our beta gets the hydroxyl, and then alpha gets what? Hydrogen, because you go from having one hydrogen to having two hydrogens. So go back and add that to your notes in the beginning. So the beta carbon gets the hydroxyl, the alpha carbon gets the hydrogen. Okay, so then we go through a second round of oxidation. Anytime you go through an oxidation step, we're going to generate an electron carrying molecule. So in step one, what did we generate? FADH2. So you start with FADH2, then you're going to make NADH. So now if we oxidize, what happens? In general, when we oxidize, we lose what? Lose hydrogens, right? When we lose hydrogens, what do we form? A new double bond, right? But now, what's the really great thing about this double bond? It's not a carbon-carbon double bond. It is a carbon-oxygen double bond. And look, do you see how these now look structurally similar now, right? Okay, so now we're in the perfect condition that if we cleave off what's in red, we'll basically have what we started out with. That's why we have to go through all of these steps so that you can do another subsequent cleavage event and another one and another one and another one until all you have left are 2 carbon units, right? Okay, so our very last one is a thiolysis reaction where we're going to cleave, if I could erase this, we're going to cleave right in between the alpha and the beta. So our beta carbon becomes carbon number 1. Right? So now what are we left with? Well, we had our 2 carbon unit attached to coenzyme A. What is this? Acetyl-CoA, right? So now we've generated our very first acetyl-CoA molecule, and we come in, bring a new CoA, hook it onto the end of our whatever's left of our fatty acid, and we can keep going, right? Okay, pretty good. Okay. All right. So if we look at a 16 carbon long, so this is right, 16 carbons. Yep.
Does it go into the thiolysis reaction of the anhydride? Like, does it show the mechanism for the thiolysis?
You don't have to know it. You don't have to. I think the book gives you some detail, but like you don't have to. You don't have to know the mechanism. All right. So if we look at something really long, something like 16 carbons, right? You need to be able to look at the structure and know where the different sites are, where you're going to have thiolysis occurring. If I ask you, if I say, okay, here's a fatty acid, draw me the product after 3 rounds of beta oxidation or whatever it is, you need to be able to do that. So you need to be able to understand those 4 reactions and predict what products are going to be made. Okay, if we go through 3 reactions, what are we going to make? What are our end products going to be? So just 3 reactions. This is showing all 8, but what happens if we go through 3? You're going to have 1, 2, 3 cleavage events. So this is all going to be attached to S-CoA, right? What else are you going to have? This is going to be 1 acyl, this one, and then this one. So you're going to make how many acetyl-CoAs? 3 acetyl-CoAs. Okay, what else do you get? 3 FADs, 3 NADHs. Okay, so just make sure that you can pick that out. Or if I give you a 12 carbon long chain, how many rounds of beta oxidation are you going to undergo? What are your products going to be? Right? All of that kind of stuff. So the other thing, yeah.
So like how the cleavage reaction is different from the acetyl-CoA, like you have 7 cleavage reactions for 8 acetyl-CoA. So when you say round,
That's one round is 1 cleavage event, right? You go through all 4 steps and you end up with 1 cleavage event. Yeah, so right, what we're just showing is where the last step cleavage occurs. The last step cleavage won't occur unless the 3 previous reactions occur.
you have 3 rounds, you have 4 acetyl-CoA.
You would have 1, 2, 3 cleavage events. You wouldn't have 4 acetyl-CoAs because you have this end product. You have 4 products. 1, 2, 3, 4.
Okay.
You would only have 3 acetyl-CoAs. Then you would have a fatty acid that would be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 carbons long.
So you want to make sure it's specified.
Because if you just said it was 4 acetyl-CoAs
Or 3 acetyl-CoA
and not the, yeah, don't leave out the other products. So draw it out. That's a really good thing. Draw it out, right? Even if you just do the skeletal diagram, you know, you don't have to show all the carbons, but absolutely. Okay. So, oh, the other thing that's really cool about fatty acid oxidation is that it also makes water. So it's a good thing in order to be able to generate water. All right. So if we look at what's going to happen when we make NADH, we make FADH2. What's going to happen? Are they going to enter the citrate cycle? No, they're going to enter directly into electron transport chain. But do they generate the same amount of ATP? No, they don't. Why not? The thing. So where do they enter the electron transport chain? Okay, so FADH2 enters at complex 2, so it skips complex 1. So you have fewer ATP made. How about NADH? Enters at complex 1, so we get the maximal ATP, right? And I would always give you those ratios. I'll give you the ratios, but I won't tell you which complex it comes in at. But I'll give you the ratios. Remind me. If I forget, just remind me and I'll write it on the board. Okay. Yes.
Just going back to what you were saying about the 3 rounds. So when there's 3 acetyl-CoA, would it also be the 3 NADH?
Yes.
Okay.
Yes. So every time you do a round, that's great. Every time you do 1 round, right? What do you make from 1 single round? for the first reaction, you're going to generate 1 molecule of FADH2, right? Here, okay, I haven't generated anything. Here, I'm going to generate an NADH, and then here I generate the Glycolysis, so I generate the Acetyl-Coa. So every time I go through, I make 1 FADH2, 1 NADH, and 1 Acetyl-Coa.
But if you do the whole chain, you're always gonna have one more acetyl-CoA than you have in ADH and FADH.
Exactly, because of that last cleavage reaction. Because in that last cleavage reaction, right? If you look here, right? So let's just say we go through all the rounds, right? So let me erase this. So how many different acetyl-CoAs, you just kind of circle them and you can count them. We do this, right? So that last cleavage event is going to give us 2 acetyl-CoA molecules, right? Instead of just 1. So if you count, that's 1, 2, 3, 4, 5, 6, 7, 8 acetyl-CoA units, but count how many cleavage events occurred. How many of the red tick marks do you have? 1, 2, 3, 4, 5, 6, 7. So you're only making, because you had 7 cleavage reactions, 7 FADH, 7 FADH2, sorry, and 7 NADH molecules, right? So down here. But you actually make 8 acetyl-CoAs. That's important. Yes. Good. Okay. All right. So we're generating ATP just from our NADH and our FADH2. But then talk about acetyl-CoA. Can we relate acetyl-CoA to the citrate cycle? Absolutely. So you have to go back and remember, right? Usually when we talk about the citrate cycle, what are we talking about for the citrate cycle? We're talking about 1 molecule of glucose coming in. 1 molecule of glucose coming in is going to give us what? 2 acetyl-CoAs. So we're kind of used to thinking in terms of 2 acetyl-CoAs for the citrate cycle, right? But here, how many have we made? We've made 8 acetyl-CoAs. So 8 acetyl-CoAs are going to give us 24 NADHs, 8 FADH2s, and 8 GTPs. GTPs translate perfectly to ATPs. Remember that your FADH2 is going to generate less ATP than your NADH, right? And I would give you those ratios for a total of 108 molecules of ATP per 16 carbon chain. That's pretty awesome, right? Correct. Correct. You have to have the precursor for citrate. You have to. Yeah, absolutely. So just like down here, I would give you these ratios. They're here. Right? Okay. All right. So what happens, right? What happens after you have cleaved your fatty acid tail? What are you going to get? So every time you undergo that beta oxidation, right? This is that link to the electron transport chain that we talked about, right? So when we undergo that oxidation reaction, this is the ETF is what has the FAD molecule in it, and that FAD converts to FADH2. The FADH2 is picked up by an iron sulfur center inside the ETFQ oxioreductase. Woo. Then Q picks that up, right? So remember how we were talking about all the different places that Q can get electrons when we were talking about the electron transport chain last semester? You remember that? Maybe a little bit. Go back to your notes, right? But Q was kind of like the last place where you're picking up electrons in the electron transport chain. And then we transfer those from Q to complex 3. Yeah. Okay.
So that doesn't go with the comments, right?
It goes into the ETFQ oxioreductase, which ends up being almost the same as complex 2. Because where does complex 2 transition its electrons to? Q. So it's basically the same thing. Yeah. Okay. So So let's talk about, before we do that, let's add a page. I want to compare ATP production in glycolysis versus beta oxidation, right? Let's do that real quick. So if we're going to look at beta oxidation, right? Let's do the same fat that we were talking about before. Acetyl-Coa, right? So how many carbons? This was 16 carbons. So if you have 16 carbons, what are you going to make? Seven NADHs. You're going to make 7 FADH2s. What else? 8 Acetyl-Coa. See? Right? Okay. So now these 8 Acetyl-Coa are going to go into the citrate cycle, right? Citrate. So citrate is going to generate from those 8 Acetyl-Coa what? So we'll say 24 NADHs. I'm just putting them, kind of lining them up. Okay. Then what? 8 FADH2s. Okay. And then what? And we'll say 8 GTP. Woo, not GTA. GTP. Okay. All right. So if we know we're going to get from these GTP what? 8 ATP, right? Okay. So from our NADHs, total up your NADHs, what do you get? 24 plus 7 is 31. So you get 31 NADHs. Use your conversion on the bottom of the previous chart. So if we go through the electron transport chain, oxidative phosphorylation, right? How many ATPs are we going to get per NADH molecule? 2.5. Okay. So what do we get? 70. Was that 70? For 1, 2.5. 77.5. Is that right? Okay. So 77.5 ATP. ATP. Okay. So then FADH2, how many do we get per? Oh, we got to total them. I'm sorry. 8 plus 7 is 15. And we get 1.5 per. So times 1.5. That's supposed to be a 15 there. What do we get? 22.5 ATP. Why do I want to keep doing that? Okay. So how many do we get? 8 plus 77.5 plus 22.5 should give us 108. But think about, this is not net. This is total, right? What do we use ATP for? Priming. We use it for priming. So we need to subtract out our 2 ATP from priming when we went from our fat to our fatty acyl-CoA, right? So we really get 106 ATP. But that's still pretty good. So that's from a 16 carbon fat, right? So how does that compare to glucose? Well, how many carbons are in glucose? 1 glucose is 6. So how many do we need to make 18? 6, 12, 18, right? Times 3. So that would give us the same amount. So about how much ATP do we get from 1 molecule of glucose? Like 30 to 32 ATP, somewhere in there. Why can't I keep the T? So times 3 would give us 90 ATP. So we do make significantly more ATP and beta oxidation than we do in glycolysis. It's about 33%, something like that. It's not mega, but it is a difference. It absolutely is a difference. And the other benefit, right? This is one of the benefits, that you're going to get more ATP. And this is just to prove to you that I'm not lying, that the math actually works out. I kind of think y'all need that sometimes, right? Nice. Is it? So the bottom, I just said, okay, so I wanted glucose. I want to compare glucose to fats, but you can't compare apples to oranges, right? I need to compare apples to apples. So if I had a 16 carbon long fat, I need to have a 16 carbon long glucose. Well, glucose doesn't get 16 carbons long. It's only 6 carbons. So to get to 18 carbons, I need to multiply glucose times 3. 6, 12, 18. Okay, that's 18 carbons is 3 glucose molecules. So if I know from 1 glucose I get 30 to 32, if I multiply that times 3 for 3 glucose molecules, I'm going to get the ATP yield of 18 carbons trapped in glucose. Yeah. It's close. It's close. It's as close as I can't do a whole, I can't do a partial glucose. It's not perfect. Yes.
Explain why some books say 36 instead of 30 to 38. So I just read that. Yeah, we were like,
I don't know.
Okay, go ahead.
I found out, I was looking at it already, I was thinking, it's because they use different ratios. They use like, I think it's either like instead of 2.5, they use like 3 and 2.
it's like older textbooks.
Yeah, older, yeah.
Yeah, it is. It is. And you'll have to understand that all of biochemistry is a seriously evolving science, and it's evolving at a really, really rapid rate. So the more we learn, the better estimates we get. A lot of times it's really hard to estimate amount of ATP being produced in a really small organelle, right? Or how much NADH is there, how much FADH2 is there. You think as a scientist, okay, you sit down at the bench and you figure out a way to quantify the amount of NADH in the mitochondria in a given cell. That's a challenge. So we're going to learn more. And so the numbers that we are using are the most recent, most relevant. But those could change. Look, that's science. That's science. Science will constantly be changing. And as scientists, you have to be okay with that. Otherwise, you are in the wrong field. I think there could be small differences, but not drastic differences. Not anything to make a large enough impact in metabolism. I don't think that's where the difference is, person to person. That makes sense. Okay. All right. So we make more ATP per carbon in beta oxidation than we do. If you wanted to compare that perfectly, right, you could then divide that by the number of carbons, you know, but this gives us a good ballpark, right? But you're also going to generate water when you go through oxidative phosphorylation. That's your second benefit. So none of the reactions though that I've shown you produce water. Why do we produce water when we undergo oxidative phosphorylation of beta oxidation products? Where do we make water? No, we're using water. We're using water. But keep going. We're using water. And then we enter the electron transport chain, and our final acceptor is O2 converts to H2O. So we're making a ton of water because we have a lot of those FADH2 and NADH molecules that are entering the electron transport chain. And so therefore you're making a lot of water. So, all right. Okay, so remember when we talked about beta oxidation, that it had to be kind of perfect, right? Well, what happens if you have something that is trans? You have something that's cis. You have something that is odd numbered, right? Everything we did, what happened if you had an odd number? You're going to end up with a 3 carbon at the end. it's not going to work out, right? So there have to be some backup mechanisms in order to support alternative fats, right? So basically what you have to do is you have to use different enzymes, and those enzymes will process the fats into things that are substrates for beta oxidation. So it's like additional pathways that they have to go through. All right, so let's look at our first example, right? Here's our example. So this is a cis delta 9. What is this? Cis delta 12. What is that? What is that number? That's where the double bonds are. Are we counting from here? In this case, we are counting from here, but there's another way that we could count from this end. That's omega. But do you see omega? No. Okay. So just be careful about that, right? Because like we numbered them differently. So our carbonyl carbon's number 1. So you go to the first carbon that participates in the double bond. What do you notice about this carbon-carbon double bond? It is cis. In beta oxidation, if you had a fat that already had a trans bond, you're ready to go for beta oxidation, right? At some point you are. So you don't really have to worry about it. But here, if you have a cis orientation, in this case, there's 2, we have a problem. Okay, so we can process, we're going to process from this end this way, right? So our first round, where do we cleave? If this is carbon 1, this is our alpha carbon, this is our beta carbon. So where do we have a cleavage event? Between the alpha and the beta. Okay, so that makes beta carbon 1. And this would be alpha, and this would be beta. So we would have another cleavage event here, right? Okay, then we would have this being carbon number 1, alpha and beta. So we'd have another cleavage here. Then this would be alpha, and this would be beta. Is it going to work? No. So we can actually undergo 3 cycles of beta oxidation and make 3 acetyl-CoAs. And so this is the product that you would end up with after 3 cycles of beta oxidation. So then you have to have an isomerase that is going to convert that cis double bond into a trans double bond. Now you have a double bond between, this is the alpha carbon and this is the beta carbon. So now what can you do? You can undergo a cleavage reaction. You're going to release how many acetyl-CoAs? 1 acetyl-CoA, right? Now, what happens? You have, if you've cleaved here, right? What does that make this? 1. What are we going to do? Let me scroll up. Right about here. Okay, so we had our cleavage event here. We're going to release an acetyl-CoA. So we made this carbon, this is the beta carbon. We made this the carbonyl carbon, right? So we introduce a double bond here, just like in our normal process, right? But what do we have here? Another cis double bond. The enzyme won't process. Having these 2 next, they're not next to each other, but they're on carbons that are linked together. So do you see how this carbon's participating in a double bond, and then the next carbon's also participating in a double bond? That's, the enzyme won't work with that substrate. Okay, so what's going to happen is you're going to have a reductase come in, and you're going to convert that from being 2 double bonds into 1 double bond, and that 1 double bond is a trans double bond. So do you see how here this one was trans, this one is cis? Yes? The trans double bond came from the beta oxidation steps. So remember when you go through and right before thiolysis, the very last step was to create that double bond. Okay, so that's where the double bond came from. You're trying to go through beta oxidation, but you can't do the very next steps because you have another double bond on this carbon. So it like halts the process. It stops the process. So then we have to pull in a new enzyme, a reductase enzyme, that will convert two carbon-carbon double bonds into one carbon-carbon double bond between those two carbons. So do you see how that was a single bond between the two carbons? That is the one that becomes the double bond. And then we go through an isomerase reaction where we put that double bond back where it's originally supposed to be in beta oxidation. And now we can go through and cleave that double bond. Yeah. So then we can go through multiple rounds and cleave and make 5 more acetyl-CoA molecules. Yeah. Pretty good. Okay. Oh, I have this activity for us, but I don't think we're gonna have time. How about we start working on it, and then we'll answer together when we come back at the beginning of class next time. Okay, so this is all about kind of why it's energetically workable for the cell to use fats. So just for you to kind of organize everything that you need. Let me tell people on Zoom. So if you're on Zoom, let me show you where to find it. Share. Okay, so if you are on Zoom, if you go here, it's right here. What's the motivation for this? Everybody got one? So what you might want to do is what I would actually recommend doing, and I think it should be organized differently, right? So it says to pair up. One person consider biosynthesis of fatty acids and the other consider fatty acid oxidation. I would actually kind of group up and like in a group work together on biosynthesis, and then in a group work together on fatty acid, right? So, and then combine 2 groups at the end. You know what I mean? You're like, no. So like work together on synthesis, then another group's working on oxidation, and then you can work together with those 2 groups. Yeah. All right, so I'm going to end the recording because we're just gonna work on this in class, and then we'll go over answers and stuff next time. So I'm going to go ahead and end the Zoom, and y'all can work on that on your own. Okay, let's see, maybe. There we go. Any questions, I guess, before I let y'all go? All right, good to go. Oops, I thought I stopped it.